Today's lecture topic is going to be development, and specifically human development. Because we could talk about development in any kind of animal, but we're specifically caring about humans for this class. So let's start off with our definition of development. So we can talk about development as growth and change over time. So this is going to include changes that are progressive, things that are sort of advancing you, moving you forward, things like learning to talk or walk, and things that are regressive, so declines, things like declines in cognitive function that typically come along with age. We're going to talk very, very briefly about all of human development, starting in utero and continuing through adulthood and old age and on to death. So we're going to cover a very large range here. And as per usual, this is a topic that could be covered in an entire course, or multiple courses even. So we're going to keep it pretty high level here, but we're going to get a good overview of how development works, specifically as we would care about it as psychologists. As always, let's start at the beginning. We're going to start with prenatal development. Now, our textbook goes on a long-winded, rambling talk about how the process of prenatal development is fairly similar to assembling furniture, where in both cases you need to have the right raw materials. Those materials should be put together in the right order. If you are missing something, it can lead to problems. If you have extra something, you can have problems as well. If things are put together wrong or follow a different path, there can be deviations and all of those things. So we could start from that framework, but rather than focusing on these sort of comparisons, let's just look at what's actually happening. Let's look at our stages of prenatal development and what's happening as we progress through each of these phases over time. So our textbook talks about three distinct periods of prenatal development: the period of the zygote, the period of the embryo, and the period of the fetus. Now, these titles aren't necessarily consistent across stages, or across sources, I suppose you can say. The stages themselves, the 0 to 2 weeks, 2 to 8, and then 9 weeks plus, are all pretty consistent, but everybody seems to call them something different. So our period of the zygote, you might be familiar with that being called the germinal stage. And that goes from conception all the way up to 2 weeks. And that's a stage of rapid cell division. We're going to talk about each of these stages in more detail in just a minute here. Or period of the embryo, you might be familiar with that as the embryonic stage. And that's from 2 to 8 weeks, where we start growing our major bodily structures. And then the last period, the period of the fetus, is from 9 weeks right to birth. That might also be called the fetal stage, and that's where we have all of our finishing touches prior to birth. So if you've seen these under different headings, don't worry about it. They're all sort of the same basic concepts, just slightly different naming conventions. So let's start off at the beginning, as we have already begun with, and start talking about this period of the zygote. And as I said, this period starts with fertilization or conception. So this involves a sperm cell and an ovum, an egg cell, fusing and creating a fertilized egg or zygote. If we look down here at our diagram, we can see the ovary that has ovulated, releasing an egg into the fallopian tubes. In that tube, we have fertilization. So that's this stage here where sperm and egg have combined to create a zygote. And once fertilization has happened, that zygote travels along the fallopian tube, dividing as it goes, forming a blastocyst, which we'll see on the next slide is a hollow ball of cells. And that blastocyst will implant on the wall of the uterine, or the uterine wall. Sorry, the wall of the uterus would be the way to say that. So this is sort of the early stages here. And this fertilization to implantation is actually our period of the zygote, or germinal stage. So it starts when fertilization occurs. We talked about the fact that it is characterized by rapid cell division. So while that zygote is traveling down the fallopian tube, if we look in here, we can see that it starts as one cell, which becomes two, which then becomes four, which then becomes 16, and we get this exponential growth as it travels up to the point where it implants in the uterus. So as I said, that blastocyst is what we're calling the zygote once it's become a hollow ball of cells. So there's a space on the interior and the outside is sort of a dense wall of cells. And so once implantation occurs, that's the end of this period. So that typically takes about 2 weeks. So that's our 0 to 2 week mark from our list. Now, during this stage, as we're leading up to that implantation, this is when we would have twinning occur. So you might already know that there are two different kinds of twins. We have dizygotic or fraternal twins and monozygotic or identical twins. Let's start with the identical twins. So we look on the far side here, and it shows that identical twins come from a single fertilized egg. So one egg, one sperm, so they form a single zygote. And then while that division is happening, while that rapid cell division is occurring, something is going to go wrong where instead of everything staying sort of stuck together as a single blastocyst that embeds on its own, somewhere in there, this ball of cells will separate into two distinct cells. So maybe at this stage, when there's only two, maybe they lose cohesion, they'll separate out, but because these cells are ready to divide and form a full human being, we now just have two identical cells that go through the process of gestation. So our identical twins here are identical. They have the exact same genetic code because they started as the same cell. They started from the same fertilized egg, and so they're going to be identical in their genetic code because of it. In contrast, our fraternal twins, or dizygotic twins, these come from a situation where two eggs happen to be released from the fallopian or into the fallopian tubes at a time. So again, if we hop back to our diagram here, we've shown ovulation where a single ovum is released. But in the case of those fraternal twins, maybe you release two from the one side or one from each side, but somehow you've ended up releasing two egg cells and both get fertilized. So in that case, these siblings aren't necessarily identical, hence being called fraternal twins. So they don't have 100% of the same DNA that we see in our monozygotic twins, but these are simply siblings who happen to be born at the same time. So they have the same amount of shared genetic material as any siblings, which means they share about 50% of their DNA between each other. And so they can have different things, like they can be different sexes. With our identical twins, because they have the exact same genetic material and our sex is determined by X and Y chromosomes, then both are going to have the same number of X and Y chromosomes, and so both will be the same sex. With fraternal twins, you can have two different sexes. They can still be the same sex, or they can be opposite sexes. It's, again, the same odds as if we were looking at just normal siblings who weren't being born at the same time. And that's because they come from two separately fertilized eggs, so they each have a 50-50 chance of being male or female. For those of us who are interested in the genetic components of things like behaviors or even psychological disorders, having these two types of twins and understanding that they have differing amounts of genetic similarity, our identical twins being 100% the same and the fraternal twins only being 50% the same, we can actually use that knowledge to help us determine if something has a strong genetic component or not. Especially where twins, whether they're identical or fraternal, they tend to be raised together in a very similar fashion. so they have almost the same environment. That way, any differences between the two kinds of twins should theoretically be due to their differences in amount of genetic relatedness. So that is the period of the zygote. Now we're going to move on and talk about our second period, which is the period of the embryo, or the embryonic stage. So like we said, this is from 2 weeks, so after that implantation, up to 8 weeks. So let's switch over and look at the details for this one. So we said begins once implantation occurs, 2nd to 8th week post-conception or after fertilization. In this stage, we start seeing the development of some major life support structures. So early on, we see the formation of the placenta and umbilical cord. So our placenta is located on the uterine wall, and it contains a whole bunch of membranes that will allow nutrients to pass from the mother's blood into the umbilical cord to the growing fetus. So in turn, that umbilical cord is then going to carry its own blood vessels to carry those nutrients and oxygen, all those things the embryo needs to keep growing, into its body. And it also cycles back waste products. So as the cells in the embryo create waste, they're going to be excreted through that umbilical cord out to the mother's bloodstream. This fresh supply of nutrients and the ability to get rid of waste products allows those embryonic cells to divide really, really rapidly, and they start to specialize. They start forming organs and all of our systems, and by the time they hit 8 weeks, the heart of this only 2-centimeter long embryo begins to beat. We start seeing things like brain matter starting to develop, facial features start to form, and so in the image here you can start seeing the beginnings of what will become eyes. So this is the stage where things start coming together in sort of a rough form. Development during this rough stage tends to follow 2 basic principles of development. So we can talk about the cephalocaudal principle of development. This one says that development occurs from the head to the tail, or from the top down during prenatal development. So in this case, we see massive development in the head and the brain before we start seeing changes in the extremities. So if we scroll back here, we can see a lot more development in that head structure than anywhere else in the body. So we're saying that, wrong way. We're saying that development starts at the head, it starts at the top. Those structures are going to develop first, and then it's going to move down the body in terms of development. Our second principle is a very similar idea, and it says, so this is the proximodistal principle of development. So development proceeds from the inside outward. So proximal meaning towards the middle, and distal meaning towards the outside. So in this case, our internal organs will start to form first, and things in our extremities, things like fingers and toes, will develop later on. And the idea here is that the more important structures, the things that we critically need to survive, things like our brain and our internal organs, those develop first. And the things that are less essential, things like fingers and toes, can develop later on once those core properties have had a chance to develop. So these 2 principles work together in order to sort of govern the order in which things develop in a developing embryo. That is 2 stages down and 1 to go. So let's talk about the last one, the period of the fetus, or the fetal stage. As we've said already, this is 9 weeks up until birth. And this is when we're doing all of our refinements and finishing touches. So we start seeing things like muscle strengthening, systems starting to develop and refine, our eyes open by the time we're 24 weeks old, and a fetus is considered viable by the time they are 28 weeks old. Before that, they're a lot more likely not to survive birth. And we can look at our image here where we see things like the head is getting larger, the brain starts to develop, we see refinement of the facial features, and we start developing all of the grooves, those gyri on our brain that are sort of forming all of the important structures within our brain, that convoluted surface that makes us as good at processing information as we are. So, this is that refinement in all of our systems. We'll see this throughout the rest of the body as well. Things like muscle strength is developing. This is when babies are kicking and moving and using those muscles to build up strength. The fetus also starts to move into the position that they prefer for childbirth. So that head down position, that happens in this period. And of course, while they're getting larger and while they're developing, they're also gaining weight. There is a ton of really interesting research about what the fetus learns about the world outside their uterine environment at this point. So while they're in the womb, while they're at this point, they have developed some of those systems. Things like their ability to hear have started to become functional. So you might have heard that it's beneficial to read or sing to a child before they're born. This is the stage where that is important. And studies have shown that if infants had been read particular books while in utero, after they're born, they respond differently to books that they are familiar with versus books that they aren't familiar with. So novel books and familiar books elicit a different reaction, which is really interesting. This is also when we start seeing a development of recognition of parents' voices. So, right after birth, babies are able to discriminate their mother from the voices of other females. Other people they haven't heard before, and even they're able to tell the voice of their mother from the voice of their father. So, lots of indications that we learn quite a lot while in utero. And there are so many studies we could talk about there, but I'll keep it to just those few. The last thing I wanted to talk about before we move on into sort of birth and infancy and childhood is talking about teratogens. So these are environmental agents or substances that negatively impact prenatal development. So these are things that can be in that uterine environment, whether it's through the mother's body or however the exposure occurs, usually directly through the mother's body. Things like lead or mercury or radiation. Nicotine is a teratogen. And there's a lot of things to consider. So different kinds of teratogens have different kinds of impacts. The dose, so how much exposure they receive, can have an impact. The timing, so at what stage of development is exposure? Early in the pregnancy, later in the pregnancy? Is it during the zygote phase or the embryonic phase, or which? And as well as cumulative effects. So is it something that happens just once or over long periods of time? And in a lot of cases, we know that things are likely to be teratogens, that they have some kind of negative impact because we've seen birth defects in those born who had been exposed to these different factors. But it's very difficult to find conclusively the specific dose, the specific timing, or the specific cumulative effect that is required to see differing levels of problems. And that's because we're not able to conduct experiments. For ethical reasons, it is not possible to induce mutations through teratogen exposure. So we're stuck using correlational studies. We would look at babies who are born who have some kind of disorder, and we try and relate it to or correlate it with exposure to different factors. So was the mother smoking while pregnant, or was there alcohol present, or any of those things? So we can get a good idea that things are related, but the specifics of the dose, the timing, and the cumulative effects, those are a little bit fuzzier just due to the nature of studying these things. We talk about three major types of teratogens. These are the ones that we see talked about in most textbooks. Alcohol is a major one. So you've probably seen, if you've ever been in a bar, there are always posters that say, caution during pregnancy, don't drink alcohol while pregnant, all sorts of posters everywhere. And this is because of fetal alcohol spectrum disorder, or FASD. And this also includes the more extreme version, which is fetal alcohol syndrome, or FAS. And in this case, exposure to alcohol in utero can affect development of a fetus. So as they're developing, it can cause changes to the face structure, the formation of the brain, and of course it's going to cause long-term impact because of those differences in brain structure. The big example that you'll usually see talked about is thalidomide. And this was a drug that was used in the 1960s to prevent morning sickness. I first learned about thalidomide actually in an organic chemistry class because we were talking about chirality. And this information is not important to this class. It's just the idea that molecules aren't always mirrored, so they aren't always the same from the left side and the right side. And with thalidomide, one orientation, say the left side, was perfectly fine and safe to use to help with morning sickness. But the right side, that particular orientation, somehow interacted with things in the body during development. It acted as a teratogen, and it actually caused a lot of birth defects. The biggest one for the thalidomide is shortened limbs. So it was a very widespread issue until they figured out what was going on, and they had to look at how to separate those two orientations of the molecule. But I always remember that particular drug because of the example of molecule orientation. Another one that's more recent is the Zika virus. And this is a virus that causes microencephaly in infants, basically where the brain doesn't grow as large as it should be. So the head is a lot smaller in these infants than it should be. And with reduced sort of room in the skull, the brain is smaller as well. So this can be transmitted directly from a virus, so infected insect bites, or from intercourse with someone who has the virus. And I also kind of already mentioned some of the heavy metals. Things like lead and mercury can cause birth defects. Exposure to radiation can cause defects. Nicotine is also a very large teratogen. It can cause miscarriage, premature birth, low birth weight, cleft palate, and more. And this also actually raises some ethical issues as well, because there are some things where, like alcohol, you can be told to abstain from alcohol during pregnancy. But what about drugs used to treat disorders? There are particular drugs that help treat epilepsy and bipolar that are also known teratogens. So here's the ethical issue. Do you treat the mother for those potentially life-threatening diagnoses and keep the child at risk for developing those kinds of issues, or do you withhold treatment for the sake of the fetus? And there's no real right answer there. It's usually left to each individual to decide how to progress if there's an alternative. But those are things that you need to be aware of when you're looking at these kinds of issues.